I'm now going to turn this over to my colleague, Dr. Edward Li, Associate Professor at the University of New England College of Pharmacy, to address how biosimilars will impact clinical practice. Dr. Li.
Thank you for that introduction. And I'm an oncology pharmacist by training, so most of the spin I'll be giving on this portion of the presentation will actually revolve on oncology as a specific example about how biosimilars will impact what we do in clinical practice. And before we get into some of the actual impact as far as how this new regulatory class of drugs will impact us in the clinics and in our practice, let's talk about what currently are the approved products out there that this biosimilar law allowed to get approved. And we have a number of drugs, new biosimilars approved for indications ranging from the supportive care of cancer, that's a filgrastim biosimilar, to inflammatory diseases such as infliximab, etanercept, and adalimumab biosimilars, and also insulin glargine. Although it is theoretically and from a strict regulatory standpoint not a biosimilar, the data that was submitted was a biosimilar package, but it's really based off a technicality that insulin glargine is approved currently as a new drug through the new drug application process. So it was kind of classified as more this follow-on biologic, but for all intents and purposes, it was approved through submitting data that showed it was a biosimilar to its reference product. And of course, we know that to be used for diabetes. And so in terms of what our biosimilar is going to do for us in our clinical practice and how we go about treating our patients, I think the first and foremost and most important impact that it's going to have is to help to lower cost of these expensive therapeutics. These are really impactful therapeutic agents, and we know them to be very expensive. And through lowering cost, perhaps we're going to see a behavior change in kind of how we use these products, which will hopefully lead to an increase in access to these medications. And we all know that the principle of as cost approaches zero, our utilization of that particular product will increase. And this also changes our paradigm in terms of looking at the study designs and the relative endpoints in terms of how we review products for formulary consideration. It also changes our paradigm and our thinking about now dispensing and substituting these biological products out in the real world. So before we're operating in a world of substituting generic products, but this class of medications now will have us start to think about substituting biological products in addition to small molecules. And lastly, I think our role as providers is to be aware of pharmacovigilance issues, again, because of the issues regarding immunogenicity as previously described, and how pharmacovigilance really spins into different regulatory standards such as naming of biologics and biosimilars. So let's start off with cost considerations and what we think about how biosimilars will affect the overall cost of these products in the United States and how that's going to change what we do. So if we look at the top expenditures in the United States, this is our report that we publish every year in the American Journal of Health System Pharmacy. Our updated report will be published in just a few months, a month or two. And this particular data is from 2015. And if we look at the top expenditures in clinics, so those are physician offices primarily, we see that the highest utilization and expenditures would be essentially the biological products, things like infliximab, pegfilgrastim, rituximab, EPO, Bev, and trastuzumab, bevacizumab and trastuzumab. And you can see how a lot of these products are used in cancer. And if you look at the top 25 drugs in hospitals, they're pretty much the same in terms of infliximab, rituximab, pegfilgrastim, being up there, as well as bevacizumab, trastuzumab being on that list as well. And you can see that, again, the big three cancer drugs, rituximab, bevacizumab, and trastuzumab, are consistently within the top 10 of expenditures within the United States in the clinics and hospital channel. And if we were just to do a little bit of arithmetic and think about if there was just simply a 30% discount on these three agents alone, that would save us about $2.7 billion annually on this. And that's a lot of money to be using for potentially other products. And the other side of this is pegfilgrastim is ranked typically around number 2 or 3 in clinic and hospital expenditures, with about $3.7 billion spent in 2015. So there's a lot of room for improvement in terms of lowering the cost and being able to lower the overall cost of care in the United States. And the other thing that we have to think about is, specifically in oncology, the focus is now on what we call immuno-oncology agents. So these are checkpoint inhibitors that essentially prime the immune system to recognize cancerous cells in the body. We already have 3 checkpoint inhibitors approved. That's ipilimumab, nivolumab, and pembrolizumab. And these drugs, these biologics, are seeing an enormous growth in their utilization and expenditures in the United States. It's actually estimated that this whole class of medications by 2020 will reach about $7 billion in the United States. And you can see that graph on the right-hand side just escalate up. Actually, I can tell you that in 2016 it's actually outpacing this particular forecast right now. So it's probably going to be far more than $7 billion by 2020. And also, this is a focus of the pipeline as well. There's many, many drugs in the pipeline in various phases of their development. So what is our healthcare system going to do to pay for these novel therapeutics that have a favorable toxicity profile and a favorable efficacy profile as well? How are we going to pay for that? And to get the answer to that, in terms of one strategy to do that, we have to look at history in terms of how we've paid for new products before the era of biosimilars. And what we did was we looked at all of our expenditure reports going back to 2010. And you can see that the blue bars are the actual total expenditures of just oncology products in the United States, and the red line is the percentage growth from the previous year during that time period. And you can see that from 2010 to 2013, there's really not much growth in oncology drug spending in the United States. But in 2013, that's when our spending really started to escalate. And in 2014, 15, and 16, it just continues to rise. So what really happened during that time that allowed this moderation of growth? This was when generic gemcitabine was starting to become more prevalent. Generic docetaxel was approved, and generic oxaliplatin was approved during that time. Those were the 3 most expensive cancer drugs at the time, which suddenly became generic. And by allowing these, by allowing providers to have this increased competition and be introduced in the marketplace, this helped to moderate the growth of oncology drug expenditures during this time period. Whereas in 2013 and 2014 and every year since then, there really hasn't been a blockbuster drug that's actually gone generic that helped to moderate the growth. So that's what we're seeing here today in terms of why these expenditures are continuing to increase. And so here's another example with some cost savings once you introduce some competition with another filgrastim product. This is TBO filgrastim. While it's not approved as a biosimilar in the United States, the equivalent product is actually a biosimilar in Europe. So, but the whole concept of competition applies in this situation, where if we looked at a 1-year period of time, you could see that there's a cumulative cost savings in terms of if you just replaced in this particular analysis, if you just replace TBO filgrastim utilization with the cost of filgrastim, the difference between reality and that scenario would have been about $14 million in terms of this particular time frame. So you can see that there's significant cost savings to be achieved just through competition itself. And when you look at the real-world patterns of filgrastim products, that's filgrastim and TBO filgrastim, you can see kind of this moderation. It's starting to moderate in terms of the total decrease in myeloid growth factor spending as market share of the competitor products started to increase. And I can tell you that when you take this data out into 2016, it looks a lot better, and the spending actually has decreased even more because of the introduction of the biosimilar filgrastim products as well. Okay, so that's essentially the summary of the cost considerations and how cost will, hopefully this increased competition will help to moderate the costs in practice. And so the second aspect of how this is going to influence what we do on a day-to-day basis is the whole concept of evidence-based medicine and understanding this extrapolation paradigm. And to do that, we have to look at first at kind of how the biosimilar pathway compares with the branded or standard originator registration pathway, right? And remember that the originator, when a new molecule is approved, the purpose of the regulatory approval pathway is to demonstrate benefit, demonstrate efficacy and safety of that particular product. So in that situation, yes, analytical studies are going to be done, preclinical studies and clinical pharmacology studies, PK/PD studies are going to be done. But really what we focus on as clinicians and where the data is extensive in its development and resource utilization to get it approved is under the clinical studies, right? And as clinicians, we spend a lot of time discussing these clinical trials. We do a lot of journal clubs with our students and our colleagues, and we talk about the clinical studies a lot in order to inform how we're going to use that in our patient population. However, with the biosimilar pathway, the regulatory approval is not to, the purpose of the regulatory approval is not to establish benefit in terms of efficacy and safety, but the purpose is to establish biosimilarity and how this molecule, the biosimilar, compares to the originator molecule to ensure that there are essentially no clinical differences between the two products. And so you can see that the paradigm is much more focused on the analytical part of the data package, the structure and function, the preclinical pharmacology assays, the clinical pharmacology program. PK/PD studies are going to be important as well. And the clinical studies are actually the least important or the smallest triangle as part of this pyramid because that simply is the cap of the data package, the double check that's conducted in a sensitive patient population using sensitive endpoints simply to this study is designed that if there was a difference between the two products, we would be able to detect that difference in a sensitive endpoint in a sensitive population of patients. So again, the exercise in those clinical studies is not to demonstrate safety and efficacy. It's to demonstrate that there are no clinically meaningful differences between the biosimilar and the reference product. What's a good example of this in the oncology world? Well, a good example is for something, say, of trastuzumab and biosimilar trastuzumab. So if you can think about kind of, how we get to the long-term outcome we want, which is disease-free survival or overall survival with trastuzumab, when we administer the biologic, we expect a pharmacologic action. And we expect, it's not on here, but we expect a certain level of PK/PD metrics, and that informs our short-term outcomes, which is response rate, which will then translate into the long-term outcomes that we want to see. And so we know what the pharmacologic action of reference trastuzumab is, and we know what the response rate is, and we know what the kind of survival data is with reference trastuzumab. That's all been well documented. So the biosimilar trastuzumab then, before it's even administered to patients and and developed, it's developed through that reverse engineering, again, concept where the structure and function will be highly similar to the reference trastuzumab. So its pharmacologic action will essentially be the same as reference trastuzumab. Again, it's not on here, but the PK/PD studies are done to inform that those are essentially going to be the same as well in terms of pharmacokinetics and pharmacodynamics. So the clinical comparison to again confirm that there are no clinically meaningful differences between biosimilar trastuzumab and reference trastuzumab is that response rate. And if that's similar to each other, then there's really no reason to believe that there would be any difference in the long-term outcomes that we want to see, which is survival, such as disease-free survival and overall survival, right? So again, the purpose of the regulatory exercise is to demonstrate that comparability, not to demonstrate safety and efficacy of the product. And so that's one specific example in one specific indication. But again, if a biosimilar is going to be approved for all of the reference products, FDA-approved indications, there has to be some sort of justification to allow that indication to be in the product's labeling. And going back again to the concept of this exercise is not to demonstrate safety and efficacy. The data package to allow the FDA to approve the biosimilar for all of the reference products indications is based off of a concept called extrapolation based on scientific justification. So unlike the reference product, the biosimilar doesn't need to conduct a clinical trial for each indication. And the reason for this is because if the structure of the biosimilar is the same or is highly similar to the reference, as is the biological function, as is the clinical, the non-clinical studies and the toxicology studies, as is the human PK/PD studies, and again, all of this is submitted to the FDA for their review. And there is a clinical study in a sensitive indication. So what sensitive means is that if there was a difference between the biosimilar and reference product, you'd be able to pick that up in that population. So if that's highly similar, then there's really no reason to believe that you can't justify using the biosimilar for all of the other reference products indications as well. And again, that's based off of the scientific justification based off the totality of the evidence that includes the physiochemical studies, the functional characteristics, the PK/PD studies, and all of the clinical data as well. So what does this paradigm do for us, and how does this affect us as clinicians? Well, remember, when we do our formulary reviews, we are looking at those randomized phase 3 controlled trials that demonstrate safety and efficacy for a particular indication. However, when we review biosimilars for formulary consideration, we're not going to see those the same phase 3 randomized controlled trials that the reference product had. Again, because it's that clinical comparison using an adequately sensitive endpoint in an adequately sensitive population. And so that endpoint may not be the clinical endpoint that we want to see to demonstrate safety and efficacy. Again, that's not designed to look at that. That's not the purpose of the biosimilar exercise. So this really changes. So if we were married to the evaluation of these randomized phase 3 controlled studies, we're really going to think that biosimilars then don't have the same level of data, right? So it really has to change our thinking in terms of what data do we need to look at for formulary consideration. So the data package that we should be looking at is pretty much the same data package that the FDA looks at, which is the totality of the evidence. So it's including that physiochemical features, functional studies, PK/PD, and also those clinical comparisons as well. And I think the FDA briefing documents that's submitted and publicly available online will be very helpful to look at when we do our formulary considerations as well. They include their interpretations of the evidence and whether or not the FDA staff believes that it actually is a biosimilar, and to what extent there is similarity between the two products. So those can be really helpful resources to look at when we look at these products for formulary consideration. The other aspect is we should also focus on those non-clinical considerations as well, because again, to be approved as a biosimilar, these products have to be not clinically significantly different from each other. So we're going to focus on those non-clinical considerations, such as the cost of the product and which product that we can get for a better cost, the product presentation and user interface, so how this is handled. So is something actually formulated and presented to us in an auto-injector, a single-use syringe, or a multi-dose vial, and what's going to be the most advantageous to our practice in terms of using it in our patient population. We're going to think about storage and stability. There can be, within limits, some differences in storage and stability between the products. So there could be a situation where one product has a more advantageous storage or stability profile than the other. And the product supply and how reliable the supplies of getting these products are. We live in an era, at least in oncology, of oncology drug shortages, and so knowing that you can get an adequate supply of the product is going to be very important as well. So that concludes the extrapolation portion of that. And so the other aspect we need to think about is substitution and how biologics will be substituted in real life. And to understand substitution, we need to go and look at this additional designation by the FDA, which is interchangeability. So again, interchangeability is an additional FDA regulatory designation that requires a different data standard than biosimilarity alone. And what this means is that there needs to be a dedicated switching study that's performed looking at some endpoints like PK/PD to ensure that if theoretically you switched back and forth between the biosimilar and reference product, that you would actually achieve the same exact outcome as if you gave the reference product alone. And the impact of this interchangeability designation is that the FDA and the law actually makes it pretty clear that these types of products may be appropriate to be substituted without the intervention of the prescribing provider. And this impacts our state substitution laws for biosimilars and actually opens the door for state substitution laws to allow a pharmacist actually to substitute biological products. So again, it's the state pharmacy practice laws that gives a person like myself, a pharmacist, the authority to act independently of the prescriber to dispense an equivalent, keyword being equivalent, low cost, and another keyword being lower cost, medicinal product. And in certain states, we are mandated to do this, not just simply given the authority, but actually mandated to dispense the lower cost product. And so the framework for substitution here is built upon the same framework as for the substitution of generic drugs. So there's typically a product criteria attached to this. So there's an FDA publication that rates the interchangeability and equivalence of generic products versus their branded counterparts. And typically states use that criteria for substitution as the product criteria for substitution. In this situation, that's going to be tied to the FDA interchangeability designation for biosimilars. DAW is an important part of it. The dispense as written and allowing no substitution. So you know this as writing brand medically necessary or no substitution on the prescription. Communicating with the prescriber and the patient that a substitution is made. That's in many laws about generic substitution as well. Record keeping, keeping records on substitution is always important and not an issue in this digital age. And a lot of the state law framework includes a hospital health system exemption in that if you have a robust formulary system and P&T committee, then you're kind of exempted from this whole process. So if you take a look at some of the enacted biosimilar substitution laws out there, you can see that, yes, there is that DAW provision in all of them, which we all agree is a good thing. The substitution criteria is all tied to the FDA designation of being interchangeable. The difference really is in the record-keeping time frame. Again, not a big deal in the digital era. The prescriber-patient communication can be a little bit different. Again, some communication plans talk about informing through either phone calls or letters, but then some states allow for notification through a shared EMR. So the common elements for substitution of interchangeable biosimilars, again, is that they're all going to be tied to this FDA designation of being interchangeable. So a pharmacist would not be allowed to substitute a biosimilar for the reference if it wasn't designated as interchangeable by the FDA. And the prescriber would be able to still write brand medically necessary or dispense as written to prevent that substitution. You can do that with branded products currently, so you should be able to, branded products for generics, you can do that, so you should be able to do that for biosimilars as well. The prescriber has to be notified in essentially all of the substitution laws. Some of the laws say communicate with, but again, don't be surprised if you get a notification saying that somebody was substituted for the interchangeable biosimilar based off of this provision. And again, patients must be notified in many of the states' substitutions as law, and that's a good thing just to inform patients of what they're actually getting. There are different variations between the states. Again, each state acts independently and decides on legislation that's appropriate for their population. So again, it's not going to be a standard across the board. So all of the substitution goes into another aspect that's important to clinicians, the last aspect that's important to clinicians, which is pharmacovigilance. Really broadly defined, pharmacovigilance is risk identification, post-market risk identification. And this particular slide says biosimilar pharmacovigilance, but really you can just eliminate the word biosimilar from this and just call this pharmacovigilance in general. In that post-FDA approval, because with all biologics, not just biosimilars, with all biologics, there is a risk that we didn't identify a rare but serious adverse event before in those registration studies, those pre-approval types of studies, because the numbers are not robust enough to detect these rare adverse events. And so in terms of pharmacovigilance and being able to track these products over time to identify whether or not additional risks exist is an important thing. And we've seen this happen, and package inserts being updated based on safety events over the years. And a good example of that would be opportunistic infections with the TNF inhibitors, and kind of having those be updated over the years. And a lot of that is based off of information that is collected, that the FDA gets from healthcare providers and voluntary reports to the FDA about the risks that are identified in the broader patient population. And so once a risk is identified and characterized, that's the right-hand side of the chart here, which the FDA will start to think about a risk minimization plan. But we're not talking about risk minimization plans right now. We're simply just talking about risk identification and whether or not there actually is a difference in risk between the biosimilar product and the reference product. And so to facilitate this kind of tracking and traceability and the data reporting, there's a bunch of ways that can facilitate that. Number one is just integration of orders and robust medical records in the electronic health record. And it's going to be important to be able to track the specific product that's actually been given to patients, integrating drug codes like HCPCS codes into claims and being able to sort through those through claims data studies to identify risks. But also the naming of these products is going to be important to help facilitate proper attribution of adverse events, because really the important part of this is when adverse events are reported, that the correct product is attributed to the adverse event. So it's really not going to be good enough just to say filgrastim caused a specific adverse event, but it's important to talk about which specific filgrastim product actually caused that particular adverse event, because now we're in an era of multi-source biologics. And so to really highlight why this is important in terms of correct attribution, this is a case study that we performed on enoxaparin products, looking at branded enoxaparin versus generic enoxaparin products. So we looked at claims data to look at the incidence of hits with branded versus generic enoxaparin, and saw essentially no clinical difference between the two products in the incidence of hit. And we saw market share for the generic enoxaparin increase after the loss of exclusivity in 2010, but the chart on the right-hand side actually essentially says that a lot of the voluntary spontaneous adverse event reporting was actually labeling the branded product over the generic product as causing hits. So a lot of that is questionable as far as do people know exactly which product they were getting. It was pretty unclear in terms of having a robust way of documenting exactly was it brand or generic. And so that's why the FDA came out with some more specific guidance on how they want these biosimilars to be named. Again, to facilitate that pharmacovigilance, that if you are going to report an adverse event, this distinguisher in the name will help you facilitate that pharmacovigilance and report correctly which exact product actually caused the problem. And so this guidance basically says that the FDA is going to institute a random 4-letter suffix to all biologics, so it's not just biosimilars, but the reference biologic will have this 4-letter suffix as well. So you can see the example there for a hypothetical biologic, replicamab, so you'll have hypothetically these two suffixes there, and this allows the common non-proprietary name to be grouped together in like an electronic medical record, and really the reason why the FDA made the suffix mandatory for all biologics now is to kind of reduce that perception that the biosimilar is inferior to the reference product. So if only the biosimilars had it, it kind of has this perception that the biosimilar is inferior, when in reality it was just another mechanism to help track what product is causing what adverse event. And so the role of the provider in pharmacovigilance then is mostly to monitor and report. Some of you out there may be involved in research, specifically outcomes research as well, and will participate in perhaps those claims data studies. But for the most part, as a clinician, mostly we're going to be doing mostly adverse event reporting, the voluntary reporting to FDA's MedWatch program. And we want to avoid medication errors in that if it was intended for the patient to receive one product, but in actuality the patient received a different product, while there may be no clinical consequence of that, that still is technically a medication error and something that should not happen. So those are things that we want to keep our eye on and report. And again, it's that correct attribution of the safety event, what was ordered versus what did the patient actually receive, and then what the event actually was. And we can facilitate that through a robust electronic health records or medical records, through barcode administration, through robust medication reconciliation, and thinking about when patients transition from different settings of care, that it's well documented what particular biological product that they're actually getting, so that again everybody's on the same page of what the person is getting. So that's it, and I just want to end with a couple of tips for practice in terms of what we should be aware of in terms of biosimilars, because again, the introduction of a new regulatory class of medications is going to just change around a little bit of what we do and add onto our workflow a little bit of differences. So the first is just being aware of what biosimilar product is actually being prescribed and used, and really think about prescribing the product with either the proper name or the trade name with the suffix. So you're going to have to think about how are you going to write the prescription. If you're sending this to like a specialty pharmacy, how are you going to write this prescription? Do you care if it's been substituted or not? Just being a little bit more aware of how that prescription is written to facilitate the actual product you want the patient to receive. Contribute to local pharmacovigilance efforts, so that could be either through voluntary adverse event reporting or just participating in prospective registries if there is one. Monitor the long-term safety of that, so some of you, again, might be involved in the research aspect of it and looking at either claims data or doing reviews at your own institution. And also to just encourage the transparency in kind of the characterization of these products in terms of being aware of what the physiochemical characteristics of these products are, so that we're much more well informed of if there are any differences between the products. So with that, I'd like to conclude the presentation, and at this point, I want to thank everybody for attending, and have a great day.